Good afternoon, everybody. Welcome to the virtual seminary validation of technologies to avoid irregular flowering in 4 varieties of mango. My name is Leonardo Ortega. I am in charge of investigation for the National Mango Board, and I will be moderating this workshop. So, to start the workshop validation of technologies to avoid irregular flowering in 4 varieties of mangoes, let me introduce you our presenter, Dr. Maria Hilda Perez Barraza. She has done investigations for the Forest Agricultural INFAP in Nayarit. She's doing research in Mexico, has 42 years of experience in investigations in issues like mangoes. Her investigations are reproductive physiology, the use of regulation of growth, and she has directed many projects of investigation financed by CONACYT, Citrofruit, and Produceville, other than the mango cluster in the south of Sinaloa and the National Mango Board. As a product of her investigation, she has lots of technological development that has been spread out to producers, technicians, scientists, and students through publications like scientific articles in professional magazines and other means. The distribution of this scientific knowledge has been done through conferences and different events organized by scientific organizations, both nationally and international, and in meetings by extension in Mexico and in other countries. It has been spread out in many places like Chiapas, Oaxaca, Michoacán, Colima, Michoacán, Sinaloa, and Baja California Sur in Mexico. And in other countries with the National Mango Board supporting, she has been in Brazil, Peru, Ecuador, Guatemala, and Dominican Republic. In 2017 to 2019, the doctor was the first woman president of the Horticultural Mexican Society. She has won the award Nayarit for scientific investigation and technological, and she is a national investigator at level 1. In 2023, she was awarded the Mérito Frutícola for a postgraduate degree in Mexico. Now, having said that, we welcome Dr. Maria Hilda. Welcome, and go ahead with your presentation. Thank you very much, Leo, for the presentation. Greetings to everybody in this seminar, and I would like to thank the National Mango Board the invitation to participate in this type of workshops where we can share with the public, with a lot of the public, the work that we have conducted here in Mexico. In this case, we will be talking about validation of technologies to avoid irregular flowering in 4 varieties of mangoes. This project has been financed by the National Mango Board. Very well. As I mentioned, we have conducted these studies in Mexico, and I will tell you about this harvest in my country. There is an area of about 200,000 hectares distributed or established in 23 different states in the country. But the main states are the ones in the map in the side of the Pacific coast from Chiapas all the way up to the north, Sinaloa, and some in Baja California Sur. This area has the varieties that are used for exportation like Tommy Atkins, Adolfo, Ken, Keith, and Hayden. We also grow Manila, but this has been a type that has not been exported, but it's very relative because in the international market is consumed. But this one is produced mostly in the state of Veracruz in the Gulf of Mexico. All of this area throughout the country takes us to generates a production of 2.2 million tons. We are the 5th mango producer but we are number one in exports. And it's relevant, the areas by area: Chiapas, Sinaloa, Nayarit, and Michoacan. But because of the production per hectare, Guerrero has a higher 16 tons per hectare. We have done this work in Nayarit, that is located in the central part of the country near the Pacific coast, as you can see it here. We have an area there of almost 30,000 hectares. The varieties are Ataulfo, that's number one, followed by Tommy, Ken, and Kid in that order. And they are produced in more than 320,000 tons are produced. The varieties are exposed to several problems, including them the health, mainly illness like prognosis, where they are very susceptible for different kind of illness, plagues like flies, that we have to have treatment, quarantine, so that it can be exported, and other problems, Mango Niño and Ataulfo, for instance, can, which is the hardest of all to get it to flower. But one of the main ones is the uncertainty to the environmental condition. So the dependency on these varieties to the changes in climate, because that causes low productivity in the last few years for these varieties. For instance, during the flower differentiation, when the buds need to differentiate, if it's too hot on the ground, the irrigation, the flower will be irregular, and this will be affecting Tommy and Ken not the most. El Ataulfo much less, but it can manage to flower some, but it's not enough to have a commercial harvest that would be sufficient. That has caused issues with production and export. But when we have cool autumns during the flower induction, we have the whole evening, lots of flowers and very good harvest. This carries problems because it concentrates the problems. And the main problem in the country, it's located in the states of the central area like Nayarit, Colima, Jalisco, etc., which are the ones that we are there with the four types. This production that is concentrated increases the amount produced and lowers the demand and lowers the price in the market. Nayarit is one of the markets that's most affected by this. The climate change, which is the climate variant that is now more regular than in prior years, we cannot regulate that. There's nothing we can do about it, but we can characterize it. We have to know, we need to understand which factors of the climate change, temperature and humidity. What is it that affects us with the mango? What are the processes? With regards to that, or we can study how can we mitigate the effect that could be done through an integral like pruning, nutrition, irrigation, growth regulators, and even or more than anything those that inhibit the gibberellin and application of hormones that we have studied like gibberellina, and that's another issue for delaying. And with this integrated work, we can have constant staggered productions. The first event that is very susceptible to those changes in temperature is the transition from a vegetative to reproductive state. And this is regulated by the environment, from the soil, climate, how nutrition, pruning, use of regulators of growth, and the metabolism of the tree, the growth hormones, genes, And depending on the conditions where that is developed, this also regulates the process, which is very important. Within the main, the temperature, for instance, must be cool between 15 and 10 degrees during the day and 10 and 15 degrees at night in those areas that are subtropical, so that the flowering can be induced. And we have a good differentiation. But in the tropical areas, lots of authors mentioned here say that this type of temperature does not exist. Rotation and age are more important in rotation between 5 and 6 months of age, but through other studies in Nayarit, Mexico, in tropical areas, we have observed that when there's no temperatures during the differentiation period below 20 centigrade, it's a problem for flowering. So in the tropics in Nayarit, we need between 18, 15, 19, 14 degrees of temperature at night. And the rotation is not so important because with the temperature that we have during that period, there's growth up to 4 weeks, and we have managed to have them flower. In order to modify the flowering process, we need to know it. We need to study closely the trees, each one of the varieties. What is the flowering? It's a multiple stage process involving from the transformation of a bud from a vegetative state that generates leaves, and to instead convert it into a reproductive that would generate the flowers in the case of the mango. This step is very important because it's done through different processes. Induction, that is done through an environmental signal initiation once the bud has been induced, and then the floral differentiation, the whole formation of the flower, so that the tree can start with flowering and flowering, which also depends from the environment. What is affecting us? We start from a very complex the meristem flowering. There are some models the professor had come out with it, and it's hard to understand, but I have explained here that what does a meristem, what causes the meristem to turn into vegetative or floral. What condition creates vegetative growth? For instance, the harvest from the year before with lots of fruit probably would generate what we call alternation, which would be lower amounts of flower because fibrilinas are acting there. And the fact that a tree produces from the beginning, the development of fertilization, it's generating lots of fibrilinas, and after the harvest, there's lots of gibberellins and lots of nutrients, and that creates a lots of vegetative growth. Excessive fertilization with nitrogen, which is one of the nutrients that are used for vegetative growth, and also depending on when it is applied, the humidity is also a condition that affects the vegetative growth. When we have lots of vegetative growths and young ones, there will be flowers with flowers, which heavy pruning would help with that. The shading of the trees, that will also take us to less flowering. So they need the humidity, and there are some endogenous hormones like gibberellins that has lots of harvest. And that it's important to control the genes that generate this, and we can repress those genes, and that takes us to flower. The moderate pruning, adequate fertilization, definitely not applying nitrogen during the differentiation of the buds because they would send us back to make it vegetative. There's a period of drought with the mango, and there's lots of controversy about that. Reduce vegetative growth and mature shoots because the mature shoots or the young ones receive the stimulus and to send the signal to the terminal unit. We have to pruning so that they can get the light that will help with differentiation. The photoperiod has lots of influence in the conversion from vegetative to floral, but in the mango it's not like that. But for some annual plants it is. As I mentioned, the cool temperature also affect which the hormones produced by the tree. If there is lots of gibberellin, it will not flower. But gibberellins increase and they can turn into endogenous hormones. It will close flowering. There's other studies that talks about the genes that induce the flowering, and we've seen the protein FT, which is the flower inducing that we were looking for. So there's lots of studies with regards to that. How can we modify the flowering and avoid irregular flowering? In the last few years, the producers have told us we don't want to do it early. We want to make certain that there are flowers. We have depended a lot the gibberellin inhibitors to growth retardants, it's PVC as known by a lot. There's been lots of good results with the mango. There's a lot of studies that improves the flowering, the fruits, and production. But this product has been seen as a contaminant. It contaminates the aquifers because if it's applied in the ground, it lasts for up to 2 years, and it's transduced into the product or the fruit. And because of that, there's other group of inhibitors, Sicosel, that has the same effect as gibberellins, and this is a stage where gibberellins are being formed, but avoids that. It causes that they will not be developed into active gibberellins. These have been tested in fruiting and ornamental plants. We also have the application of hormones like fibrilinas. Like we sometimes want the delay of the flowering, and there's been studies on pruning and nutrition. With all of these studies, we have the strategies to avoid irregular flowering and modify mango harvesting time in Mexico. The goal was to increase the productivity because we had lots of problems with irregular flowering with those changes in climate, and we wanted to avoid that flowering and modify it, the harvesting time. And for that, more than anyone, we wanted to have an alternative that would be more favorable with the environment. We did a 3-year study in Nayarit and Colima, and you can see it in the map. We work with Ulfo, Tommy Atkins, and Kent, the 3 varieties. We did lots of studies during those 3 years with regards to the nitrates mentioned that are very good to delay the pruning and the application of hormones like fibrillins with stimulants that are used and nowadays that are being used a lot, and lots of studies are being done with those to see what the effect does with the flowering, and it might be similar to patroputrazole. And we also did studies with other inhibitors different from the PVC. And what did we get? We evaluated for 3 years lots of variants to see if we're going to have flowering or not. A very important was to see the bud vegetative to flowering and we wanted to see if what we're doing generated these changes and how long it was taken. So we use a scale that we develop ourselves with the work with the National Mango Board, and we define 12 floral development scales, but 4 of them have been very useful to determine if they are having an effect in that transition, even when we don't see the flowers. So stage 1 is the apical vegetative meristem. These are anatomical cuts where we take samples over time with the bugs and check it through microscope and to see how the flowering is developing. And we did it in every test stage, taking pictures of the appearance of the outside of the bud. And this is what the apical bud looks like when it's starting the process of differentiation that internally we see within this. We see the bracteates that's elongated and the many stems that will let the secondary growth, and this will generate the bracteate. Externally, we see how the scales start to separate, and we see this part of the bud. Stage 3, it's a differentiated bud. They have fully grown and they're meristems. Here, there is nothing that the bud would make it back to be vegetative. If some conditions are changed, the process could be changed before, but not at this stage. At stage 4, we have the completely differentiated bud with all the floral structure well defined. And we use this to see if the change is going from vegetative to flowering in this that we're taking care of. And some of the treatments that we're doing and the results with flowering, Priscilla, PVC, Psychosil, and these are the results that we got in this investigation. You can see clearly the fast flowering, the control, and we could really use PVC to compare with the products that we were evaluating, like the inhibitors and like the control absolute or relative from the producer. There was no application of any inhibitors. What happened with this work? Well, we got some technologies that were designed to avoid irregular flowering, an alternative to PVC, and there's alternatives to PVC like PCA, uniconazole, one application of calcium PCA, 1,500 milligrams per liter, between 40 and 50 after pruning. And why do we need to vary? We need to vary that so that we could get the mango. We also had technologies within the pruning to stagger the harvest, improve the fruit hill, and improve the orchard productivity. Not the amount of production, but the productivity, the health rather, instead of the productivity. And we didn't see that it was affecting production as much when we saw that it was necessary to modify it to an earlier and the intermediate that we did 2 months after harvest. And we did that with the Ataulfo. This gave us an alternative to delay the flowering. And with that, we did increase the productivity and the price was really good. The other technologies with the use of biostimulants, and we use them to ripen buds, improve flowering, fruit set and fruit size. And it was with the application of combination of algae extracts, cytokines, amino acids, and Balmix, which is a vermicompost. And the first one is the one that had the results that were more uniform during the first 3 years. This helped us clarify the action of different growth regulators with nutrition, pruning, and the differentiation of when the flowering buds would develop, when we had the harvesting time, and the increase or not of production. We had a whole group of technologies that would take us one way or another, and we said, what must we do? we had to validate these technologies in commercials because we do investigation when we apply 8 to 10 years in that treatment. That is the best one during the study. We have to validate with the producers in a commercial producer and in a larger area. And why not in different areas of the country?
And so out of all of this came the validation project to avoid irregular blooming in 4 mangoes variety with an integrated management focus. And we're going to show you the several technologies that we tried in that study. What was the goal? Demonstrate the effect of those technologies, integrate them into the management approach, and take them to commercial farms with cooperative farmers so they could see the effects themselves. So it would not be just research. What were the specific objectives? Well, we validated the effect of the gibberellin inhibitor, basically an alternative with PBZ, but with a very similar effect. Validate the effect of early light pruning because we thought that that would actually validate our goals. We validated biostimulants with marine algae, amino acids, and cytokinins with 4 applications. So what did we do? We went to 4 commercial farms with handled by cooperative farmers, and we did so with Ataulfo, Tommy Atkins, Kent, and Keith mangoes, and we validated in half a hectare, more or less. The number of trees would depend on the density of the orchard. For example, Ataulfo was on a 10%, and we divided it 100, trees for it. If it was 100 trees per hectare, we would carry the study out in 50. Tommy had a higher density, 176 trees. Kent had 204 because the distances were shorter. And Keith was actually the youngest orchard, 7 years, and with a distance of 7 per 6, which was a pretty wide density. However, we had only considered 3 of these farms for validation, and we said, okay, let's talk to the National Mango Board and let's see if we can include Keith. But to make the budget work for us, in the half hectare of the Tommy, we're going to divide it between Keith and Tommy. So we worked a fourth of a hectare in one of these. And this is where we applied the technology. And what did we do? First, we carried out early pruning, and it was incredible work because I think we pruned practically the whole orchard, and it was early pruning. This is something that we had been testing, and we did it right after the harvest. And we took off 50 centimeters of cropping from the canopy of the tree. This cropping was around the canopy except for the Kent, where for the Kent, we just took off 20 centimeters. I mean, 50 centimeters is not too much for the Tommy or Ataulfo varieties, but for Kent it is. So for the Kent, we only took off 20 centimeters. After the pruning, we fertilized with chemical agents and using NPK and using the formulas that you see on screen. Why? Because we had research that we hadn't done ourselves, but the research was done in Nayarit by the doctor that carried out the nutrition studies, Dr. Salazar, who unfortunately is not here. But we took his studies and we decided to incorporate them in our methodology. Once we fertilized the orchards, we had 2 options. Option number 1 was after pruning and fertilizing, one single application of calcium prohexadione, PCA, around that period right after, we prune between 50 days or so in Nayarit, and that's what we had, a mature bud. And we left it at that. The other option was pruning, fertilizing, sorry, and then one application of PCA. And then after 2 months of that application, then apply biostimulants. And we did it in a right bud at the early stages, full flowering, and then fruit set. These are the products that we used as biostimulants. We used algae and amino acids, cytokinins, and crecimiento max, and in commercial doses. These are commercial products in the indicated dosage. And I was saying that we applied this on ripe bud at the initiation, full flowering, and fruit set. And we said, okay, these are the two management options. Why don't we try biostimulants by themselves? And we decided to do it in a bigger surface. So we took another area of the producer's orchard and we applied only biostimulants. And what about the other orchards? Well, we go into orchards that have 5, 6, 7 hectares, and we just get ourselves from what the producer is doing, and then obviously he prunes, fertilizes with whatever is recommended to him. And so, what did we see happening in the Ataulfo variety? In this chart, you can see the effect it had in the blooming, in the flowering timing, the percentage that we obtained, and the fruit set, which we evaluated according with the retained fruit by inflorescence. Meaning, at the beginning, there's a lot, and then some of them fall, and then we are left with 2, 1, or, well, the number depending on the variety. The flowering timing happened around 78 days after the application of PCA. What does this mean? We counted the days between the last application of PCA until the tree was in full bloom, and the tree took 78 to a full bloom. Control trees took 95 days. So the conditions were not that bad for blooming, for flowering. Even so, with these two managements, we managed to bring the flowering period from 17 to 11, but it also improved the flowering and also initial fruit set with the two management options. Now, the harvest time, the first one was May 23rd, and they harvested 80% of the fruit, Ataulfo mango. With this treatment, 75 between, but in control, they only harvested 33% because the fruit was not ready yet. Second harvest on June 7th, and that's where they got most of the fruit. In the second round, only what's left is harvested. What does this mean? That we saw about 15 days in advance, and it came early in about 80%, 80% when we applied these treatments. And when we incorporated these management treatments in the management of the orchard, we saw great results, really good results in some other varieties, because obviously this will also depend on how variety or the particular orchard responds to the treatment. And later on, I will go more into detail about what happened with this modification of the crop. We saw good results. The producer was able to get a good price, and he was very happy. What happened with the yield? We measured in kilograms of fruit per tree. You can see 64 of the control compared with the 119 that we obtained with the PCA and bio-PCA. So we're talking about a significant increase, almost double, almost an 80%, between a 70, 80% increase of the production in that orchard. And see also the weight of the fruit when we applied biostimulants. That also gave production a bit of a push. If we express this in tons, considering the numbers of trees per hectare, you can see that we practically doubled production. We went from 6 tons in control trees, we went to a 10 or almost 12 with treatments. If you pay attention, you'll see that the flowering was not that bad in Ataulfo, but in Miño was a little different, something that we didn't see here, and we did not include that variation because in Ataulfo that was not what we were looking for, but we saw that there was a lot of mango niño in these control trees, and it did well, but production was not that great because the fruit was small. A lot of them were small. So these treatments actually improved the size, the weight of the fruit. Some of the aspects that we saw when we applied PCA in Ataulfo, especially in the flowering stage, the combination of biostimulant with PCA, and then control with pretty erratic flowering. And regarding production, control barely produced any, and it was with mango niño. What happened with the Tommy Atkins variety? We applied the same treatments, and we saw excellent results by bringing flowering time earlier. What did it take the Tommy Atkins kind to reach a full flowering time since the last application of PCA until full bloom, which is what we consider 80% of the tree with open flowers? We saw that it took 107, 108 days with this other treatment, but in the control tree, it took 131 days. What does this mean? That means that we managed to push it up by 23 days or more, and we increased the percentage of flowers and the fruit set. At least in these treatments, were significantly larger than compared to the control tree. And what happened when we modify the harvest? Sometimes we had managed to even get to push it up by 30 days, but in the harvest that disappears. Why? Because the producer goes a lot by the product price. And last year was something incredible. And basically there was no price. So I mean, I don't know how the market works, but depending on the prices, then they cut and they tell us. And when they told us, they said, well, it's not ready. And they harvested in May. It's too early, even for a Tommy Atkins. And obviously they didn't harvest a lot. They just got a little bit. And until June 21st, it wasn't ready and there were no control. And I told them this is too early. And they said, well, it doesn't matter. The price is this right now, so we're going to cut. If we could harvest it whenever the fruit is ready, we would have seen great advances. It was lost because the producer cut, even though it was too green, and there's nothing we can do because we can really could buy the production of 8 trees, but buying the production of a whole hectare, that's too much. But we explained the results and they understood. I told them you have to wait, but they said, well, I can't wait. The price is this one right now. And in fruit yield, this is something that surprised them. And they said, wow, this part of the orchard is doing so much better than the others. So how was the yield in terms of kilos per tree? From 90 to 235 with these treatments. And the weight of the fruit also went up, especially with the application or the combination of biostimulants and PCA. What does this mean in tons? Well, 35 and 39 tons in this orchard versus the 15th yielded by the control. The producer was pretty happy about this. And well, I don't know how much he got for his fruit. I'm coming back to this because we saw something interesting, and it's the size of the fruit that we achieved for the tummy. In that orchard, it was very small, barely reaching export standards. And with this product, the size went up and it was noticeable. The interesting part of this validation is that the producer himself can see it with his own eyes. I can show you a few images of the flowering and the yield. Now let's talk about the Ken variety. Same same factors, and we saw that the flowering time, when they reached 80% of the tree, it happened 110 days after the application of PCA, 122 in the control tree. So 12, 14 days before the control tree. And we also had excellent percentage of flowering compared with the control tree. I'm telling you that Kent is one of the varieties with the most problems when it comes to flowering. Initial fruit set also improved vastly. There was no difference between the treatment, but with the fruit set, we evaluated around 45 days after full bloom. But that means that you lose some it, and the final set is usually just one, two fruits per tree. I mean, not all of them actually reach harvest time, but that gives us an idea of what's the yield approximately when harvest time comes around. Same when they cut the tree, when they cut the fruit off, we have to be ready. So by the harvest time, we come around and we evaluate it. And since they have to keep an eye on the price of the market by the 16th of June, they had the first harvest. They didn't harvest a lot, and then they wait until the price is good again. And in this case, they waited until the 5th of July. And again, the control trees are harvested as they are, whether they're ripe, whether they're green. They go through all the trees just the same. And obviously, we don't know how the time in particular affected the harvest because it is what it is. But the yield, the yield can see. I mean, obviously, the producer was able to see the number of boxes behind under each tree, how many kilos, and how the size and weight of the fruit improved compared to the control tree. So these two treatments, PCA and the combination of PCA and biostimulants, yielded a pretty acceptable fruit size, and it went from 105 to 118 compared to 71 kilos from the control. This takes us to a yield in terms of ton per hectare of 24 and 21 with treatment that we've included in the integrated management versus the 14 and a half tons yielded by the control trees. And I don't know the price that this would gather, this would fetch, because it's a mature project, it's a mature product. So we just wanted to see how to increase yield in this variety. Here you can see the flowering that we saw and a few images of the harvest. Finally, let's talk about Keat. Keat were the youngest trees that we saw, 7 years old in average. We evaluated the time in which they came into full bloom, and we didn't see we managed to push up flowering between 8 and 11 days compared to the control, but it didn't mean a lot when harvest came around because when we harvested the Keat, well, we know that it was going to last. We were able to push it up by 30 days or so. But what happened with flowering? We avoided irregular flowering compared with the flowering as that the control trees saw because the obviously control trees didn't receive any treatment. No PCA, no biostimulants, nothing that would favor or delay flowering. Fruit set also improved with the two treatments, PCA and the combination of PCA with biostimulants. And initial set was better. But as I said, this can change when harvest time comes around. How was the production? Well, we had from 203 to 119. And it's incredible how trees that were so young and so small could give so much fruit against, what you see here, the control tree. And take a look at the weight of the tree of the fruit. It was very noticeable with the two treatments. This led us to yield in tons per hectare of nearly 50 and between 50 and 52 tons. Absolutely incredible. The producer told me, we managed 20 and we practically doubled. And obviously he wants that. He wants that to be he wants us to do it again this year. But I said, no, this is our technology for you to adopt. And these are just a few images of the flowering and the key mango production compared with the control trees you can see below. So what were our conclusions? The combination of biostimulants and PCAs or just the PCA had a similar effect on the percentage of flowering in Ataulfo and Tommy Atkins, and we saw that in the case of Tommy Atkins, we managed to push up flowering by a lot. In the Kent and Keat varieties, the combination of biostimulants and PCA was noticeable and it favored fruit set. But both treatments also favored yield and improved the size of the fruit. So we reached the conclusion that without the application of PCA and without the application of biostimulants, flowering will still be scarce, irregular across the four varieties. But this irregular nature is even more noticeable in the Kent and Keat varieties. So it's important to combine all this technology to have the best possible result from the flowering until the yield of these varieties. What could be the benefits for the mango industry? Well, we now have an integrated management strategy that is available for transfer. And not this isn't just research. These have been validated and it can be used on the field. It's ready to be transferred into the producers in Nayarit, which is where we carried out the study. This includes pruning and nutrition and two alternatives. You could do PCA or you could do PCA and biostimulants, and this will improve fruit size and weight. And we can do this in the four varieties. This will prevent irregular flowering, and it will even allow you to push it up a little bit if you lost it because, for example, the price, the market dictates that you have to harvest early. Well, that's another thing, but we have seen that it's possible to stagger production, not stagger it, maybe within an orchard, but stagger it in the different orchards across varieties. This will improve fruit yield and size, and more than anything, it's going to improve price in those states in which the mango is punished or suffers a lot from concentration. But as we did this in Nayarit, and it has to be validated in other mango producing states, places that have similar conditions to Nayarit. Could be Sinaloa, could be Jalisco, maybe Guerrero, Chiapas, maybe not Chiapas because it's too different on the south, but at least the northern and central states in the Pacific, maybe we could validate the study there as well. What products did we obtain? we generated a series of technical charts, one for each variety. And what is this? What are these? What are these articles? We describe the technology specifically, the technology spec sheets. What was the method? What was the methodology? The results? Because if if we give a report to the National Mango Board, maybe that has like 20, 30 pages. And what we did is that we've concentrated information on these spec sheets, along with information about the study, where it was validated, what state, what other states it could be validated on, and what are the potential targets, what's the estimated cost of the technology, and what are the potential benefits. We carried out a financial analysis of the benefits of applying this technology. We included some images and some charts as well. We also included information for those who want to read more, putting the contact information for the people that created the spec sheets. And we think this is a pretty comprehensive look at what can be done, at least when it comes to the Ataulfo mango. We did the same thing for Kent with the same information, and also for Tommy Atkins and for the key variety. There's a spec sheet per variety with the same information. And I want to emphasize that this is not a guide for all mango producers. No, this is just support. This is something that we can try to do in our states, in an orchard. This is not a recipe. It's not a surefire recipe. This is just a guide. And we explain in the spec sheets what we did and the results we saw. And as I said, it's always better to validate it in other mango producing areas. Now, I would like to introduce the members of the group. Myself, Doctora Irma Julieta, Doctora Raquel Cano Medrano for physiology, and then we had two wonderful research assistants, and they are the owners of the orchards, and it's in their interest for things to work. So, but then I'm here, and that's all. Thank you very much for your attention. Thank you so much, Dr. Ilda, for the wonderful presentation you just gave us and the invaluable information that you compiled over time, both on semi-commercial as an experimental level when it comes to the validation of these technologies. We have a couple of questions, Dr. Hilda. We have a question from Dr. Victor Galan saying, how is it possible that the combination of PCA and biostimulants produce less than just the PCA? Could it be a negative interaction? He said that he saw this both in the Kent and Ataulfo varieties. How would you explain the results that you saw? Yes, from the moment we started researching, we realized that the bio products do not have an effect on flowering. The biostimulants, I mean, we're still researching them, and they have a good impact on fruit set and yield, but the PCA helps with flowering. So we think that depending on the variety, well, these are the results that we could see. But in many cases, even with just PCA, we could be on the other side. But in varieties like the Tommy Atkins, we need other products to improve the size of the fruit. And so the bio product is not going to improve flowering. It's just going to allow us to synchronize the process. But the PCAs has a good has the effect just like the Paclo of improving size. We have another question from Diego Marmolejo. How did you apply the PCA in terms of dosage and the way of application?
The proexedione is applied with the foliage 500 milligrams per liter of water, and we prepare it depending on the number of trees that we have, and it's sprayed on the trees, and that makes it so that this product is friendlier to the environment because applied to the foliage, its minimum life is like 14 days, but the other one that is applied to the crown and the molecules remain almost for 2 years. So we apply one dose of proexedione on a daily basis by spray. In one application, 45-50 days, when it's the has matured, not when it's growing. Diego Marmolejo wants to know how many years after planting can it be applied, the proexedione? Well, that depends of the goal. Proexedione has a similar action to padlock, which is recognized as to delay growth. And if I want to have trees of smaller size, we can start it in the first few years of growth. But if that's not the interest, the size of that tree is not interesting, I can wait until the tree grows and then by next year. And for Mexico, I would say after the third year in Mexico. After the fourth year, I'm sorry, because the third year is like a test, 2 years established and the third test. And after that, we can apply it if it's going to be addressed to production. But if it's going to be to maintain a small size of the tree, I think we can apply it from the first or second year of growth so it doesn't grow too much. As a matter of fact, this is one of the practices that we must implement when we want high density other than pruning, in the inhibitors of gibberellins and the use of thank you, Doctor. We have Daniel Valle has a question that what happens when we carry that floral inductions, we have the presence of heavy rain. That would be a problem. It can be that it affects in some ways the effectiveness of the product. Supposedly there should not be humidity, but that makes it harder in those places like Nayarit, where we depend a lot on the rainy season. But we have seen that if we have the adequate temperature during the months of November, the end of October, November, which is when it will be developed, the switch from vegetating to flowering. If we have cooler temperature, we get the change and we have better flowering independently of the level of humidity in Nayarit. There could be some rain, but we've had those temperatures, and much so if we apply the inhibitors with enough time. But it depends on the amount of rain, but that could affect it. Daniel has a question with regards to applying paclobutrazol. What is the recommendation of this application to be carried out? Would it be better with fractional application or one application at once? We've done just one application and the amount is 0.5 grams to 1 gram of the active ingredient Atulfo. It works with up to 0.5, 1 gram of active ingredients for every lineal meter from the top of the tree. We make the conversion with the commercial product and we apply that on the ground, but not so much, but on the trunk about 60 centimeters from the ground, and that's the only application we use. I remember that in Oaxaca, they asked that very same question. and sometimes they would see that it wasn't going to work because there was vegetative growth, and what it would do is they would do a second application. Italo, the doctor in Brazil that has lots of experience, and I agree with him that you can split it. The amount, if for every tree you're going to apply maybe 20 milliliters of commercial product or 15 or 30, you can split it in half and then apply it one time. And depending on the result, you can apply the second, but it depends on the dosage used, should be what is recommended. If you do 20 because recommended and 20 again, that would create lots of problems. And we have seen that in Brazil, where the trees sometimes don't even know whether to flower or vegetate. So we have to be very careful with the dosage. You can split it as long as it's no more than the recommended dosage and split it in two. Giorgio Battistini has several questions. When can we get those publications or technical sheets that you mentioned? Through the National Mango Board, you could distribute it, I think. I gave it to you as a product of this validation of this work, the National Mango Board. They can get it there. And my email is also listed there, the official and my personal email, and I would be more than happy to send it to you. But through you would be, because there's a lot of link to lots of people, but throughout Mexico, I'll be more than glad to send it to. We will be posting this information in our webpage. They are not there yet. We wanted to complete this workshop to then post the recording and the sheets, the fact sheets that were posted. Another question is, how many days after the pruning is recommended to apply the inhibitors of gibberellin? I don't want to talk about PVC, but we applied that propaxilline after several years, just one application, 45 to 50 days later, when we see that the budding was mature, and during that time that it takes to mature in Nayarit. And we did 3 other applications with propaxilliona in 500 dosage, so 3 applications, a total of 1,500 milligrams per liter, and we were splitting it since the buds were young, and then by the time it was 15 centimeters, 15 days the first day, 15 the second. Why every 15 days? Because it's the shelf life of this product, and we saw that with just one application, we had results, and it was much more economic. Those applications were done on the foliage or the soil. Foliage. They are on the foliage. All the inhibitors that we evaluated, alternative to Paclov, are applied on the foliage. Just the Paclov is the one that is applied as a control applied to the ground. The others are foliage. They are sprayed on the foliage. We would like to thank Dr. Ilga. We also would like you to participate in the future workshops that we have. We have one for March 19th with Dr. Stanley Friedman with regards to the incidence and gestation in the witch's broom in mangoes, and we ask you to participate. Once again, thank you to Dr. Maria Hilda, and this is the end of the workshop, and we see you in the future. Thank you, everyone. Thank you, Dr. Maria Hilda. It has been a pleasure for me to share this information with all of you.